PhD student from Washington State University. My dissertation primarily focuses on phenotypic plasticity and polyploidy in invasion using our model system of the New Zealand mud snail, but my talk sort of kind of doesn't have anything to do with that. Instead, I got interested in this comparison of mechanistic and correlative models to analyze distribution and performance. And so I kind of have to talk a little bit about distribution at first. And I've been interested in distribution for a long time. I used to work at the National Park Service in the States. I was always kind of intrigued by why some species could just seemingly take over and invade any habitat, and others couldn't. Why can they do this here, but not other places? And this has been a long-standing question in ecology dating back early to the 1900s about why organisms live where they do. And there's a lot of reasons and a lot of things that can impact where organisms are distributed. And a simplified version of this looks like this flow diagram, where extrinsic and intrinsic factors, so extrinsic, things like the environment, temperature, precipitation, and intrinsic, like some of the things Will and Sally talked about, like growth rates or polyploidy, which I look at, these things can influence an organism's performance. And environmental characteristics and the intrinsic characteristics can be lined up, and you can figure out all the possible permutations and how well the organisms perform, and you get a very basic definition of the ecological niche of the organism, or the sum of the habitats and environmental locations where an organism can successfully establish and grow. And according to ecological niche theory, this niche, or the places where they can grow, then should determine the distribution of the organism, because you have all of these possible combinations and where they can grow. And so I happen to like this as an evolutionary biologist by nature, because we can look and find where organisms live, map the environmental characteristics, and develop some kind of scheme to ask evolutionary questions. And I tend to be interested in evolutionary questions that are sort of controversial and tend to have both yes and no answers, and I wanted to kind of answer these in a new way. And so I got interested in these distribution models and environmental niche models. And you can ask cool evolutionary questions, such as things about polyploidy, like how does evolution of polyploidy affect ecological performance? So if these orange dots are diploids, and these purple dots are, say, triploids, you can map where the suitable conditions for each ploidy is, and then ask the question, well, should their distribution change or should it not change? You can also do things with climate change, so maybe temporal differences, maybe these are the same sites geographically, but their environmental conditions change temporally. And you can ask, how does climate change affect the ecological niche? There's been a lot of work done recently with niche conservatism or niche evolution. And so I'm trying to steal all this work ecologists have put into distribution models and use it to these old evolutionary questions that seem to get answered the same way. When I first started my PhD and I asked these questions, they said, oh, this has been done already. Because we go and we take an organism, we find some important variable like salinity, we bring it back to the lab, we grow it up in a bunch of containers, we measure how well they grow across salinity, you get this growth curve, right? And you can say, okay, well, the highest lambda, or this highest fitness, is in the middle, moderate salinity. Ta-da, we answered this question about where the organism can live. I wasn't really happy with that answer, because I think there's more than just one or two important environmental variables. Plus, all of these mechanistic lab studies tend to have a high cost. My first chapter did this, and I got really tired of measuring 600 individuals every single week in the lab. And I wish there was some kind of better way. And so I was interested in more broad-scale kind of applications, more correlative models, where you might have heard these called environmental niche models or species distribution models. I'm going to use them interchangeably for this talk. But the idea with these correlative models is you take a whole bunch of environmental variables and you use a model to build this idea of the niche, predict performance, and then you can build these nice, pretty maps where you can figure out, well, okay, the organism should be highly distributed here, not so highly distributed here. And there's been kind of a little bit of debate on which you should use. Anybody who's thought about, mechanistic versus correlative. But I like that if you line up all of these, the theory of what should control distribution, to how mechanistic studies work, to how correlative methods work, they kind of have the same pattern. You start off with these important variables, you figure out how they perform according to those variables. You discover something about the niche. In the case of mechanistic, you learn about the fundamental niche, which is how the organism performs across one variable and its entirety, whereas the correlative, you learn about the climatic niche, which is where the organism performs just in the spots that you know. And you have this ultimate goal of predicting distribution. And so this is kind of the tried and true way, and ecologists, especially field ecologists, have spent a lot of time and effort into modifying and perfecting the correlative method. These correlative methods, the species distribution models and environmental niche models, are published hundreds, if not thousands of times per year. They're used for a wide variety of miraculous studies, like using these models to predict where you can dig up mammoth bones, to how and where you should release endangered species. And so these models are very effective in their end result. But we're still a little skeptical of how well a correlative model can actually work. Whenever I think of correlative, as opposed to mechanistic, I cringe a little bit, because I don't actually know what's going on in them. And so, one study inadvertently almost compared these two things. And they built a species distribution response curve for 3 species, so species 1 is the green, species 2 is the gray, species 3 is the red, across temperature. And they found out that they have these kind of response curves. Then they brought those 3 species back into the lab, and they did the exact same thing for temperature. And not surprising, they don't exactly agree. But that's comparing lab studies to these species distribution models. I want to know, how well do these things actually compare to how the organism works in the field? Right? Can we use these models to actually predict how the organisms are going to do in the field? And does it compare anything to how the lab works? And so what I did is I went to Yellowstone National Park in Wyoming, and me and my colleagues, we mapped a bunch of different environmental variables. I work on a marine or a freshwater organism, so these are all water characteristics such as calcium levels. It's a mollusk, so calcium is really important. Minimum temperature, maximum temperature, conductivity, all sorts of different kind of water chemistry that we expect would be important for a marine organism. I then built a species distribution model, which this is an invasive species in Yellowstone, and figured out, okay, for all these different spots that we went to, according to this model, which should be the most heavily abundant areas? Where should the snail have invaded the most? Where should the snail have invaded the least? And then I can compare it to how they actually perform in the wild. So we set up growth cages in Yellowstone and measured how the snail grew and reproduced in these exact same spots. And we're trying to figure out how well these agree with each other. And like Will had said before, I think invasive species have a unique role in that we can look at these major ecological processes very easily and very quickly. There's a lot of abundant data on invasive species, and we can use them to answer these broader scale questions. And so my overall project idea was figuring out how well do these two things agree. And so for the field experiment, I went there, we set up the cages, we measured length, reproduction, and individual biomass over a few weeks across different size classes. And what I found out, which you don't have to memorize any of this, is that I was able to figure out where the snail, this invasive species, should be the worst, where it's the most abundant, where it reproduces the best, where it's the nastiest. And I ranked all these different places as the average ranking for the highest biomass, the highest reproduction, and the highest growth. For the distribution model, I built a map using the exact same characteristics. So we did a huge survey of Yellowstone. That was my environmental layer. I didn't pull anything from BioClimb or any of these big databases. This is the actual field measures when they grew, or when they were growing in the cages. And I used a MaxScent model, which has been shown to work on these small scales and for limited sample size, since a lot of these distribution models usually have thousands of data points, and we're working at a very, very small scale. And I was able to do sort of the same thing. These models produce something called habitat suitability indices, which can be interpreted a couple different ways, both as the probability that an organism is found at that location and how well the habitat should be suitable for the organism. And so we're getting analogous results here. Our field measures show us where the organism does the best, and our model predicts the same thing, which places should be most suitable. And so I was able to come up with a ranking system for all of the different sites that we went to. And so there's some of these are tied here, so that's why it's 9 to 14. But we see an interesting pattern. So we're able to very easily predict the best and the worst sites. These sites actually don't have any populations there, and our model correctly predicted that they would be the worst. However, these middle values seem to be jumbled up. If you take the time to look at this, it's actually not as jumbled as it looks. There's a couple of these that are ranked 2 here and ranked low here, but most of these only switch 2 or 3 rankings. And so while the middle is jumbled up a lot, it is actually a very good indicator. However, this is not really what I'm interested in. I'm more interested in how well do we actually predict the field performance. And so if we look at habitat suitability produced by our model and a measure of biomass, we see that there's a positive correlation between our model's ability to predict how suitable each site is to how well the snail actually grows in this site. So for biomass, the higher habitat suitability for each site, the more biomass the snail will put on per day. Likewise, for length, same story. A good positive correlation between habitat suitability produced by the model and how fast the snail grew in the field. And the same thing for reproduction, for how big their brood was over the course of the summer. We found a positive correlation between habitat suitability and brood size. And so these niche models are actually pretty good at predicting in-field performance for organisms. I've only been able to find one other study that sort of looked at this, and it was in birds and nesting sites. But we can see that you do produce analogous results. And for comparison, for management purposes, I'm not sure how good it would be for management, since you saw the middle values for all the rivers and streams were a bit jumbled, and you probably couldn't make very good decisions at this scale, this lake compared to this lake for invasion or for management plans. But we do see significant positive correlations for length and reproduction in regards to how well the field performance compares to these models. And so even though these models have been worked many times and have been shown to have, to be able to predict different things, we show here that it is actually a good measure of the actual performance in the field, even though it's a correlative model. Thanks. You got time? 2 minutes. Perfect. Questions? Making it easy for me.
Okay, so one of the issues with these species distribution models is you're sort of making an assumption that the species has sampled all of the different types of habitats.
Absolutely.
So you could, maybe draw connections, say this is the lake where we should expect to see the most types of snails or whatever, but it might just be, something that has to do with how the populations were sampled in the environment. So, sorry, I guess it's not a well-formed question, but do you have anything to say about that?
Yeah, so we looked at that a little bit. So we included true absences. So we had places in the park that didn't have any sales. And I also made sure that we do a lot of work on this organism. And so one of the perks that we have is that we've worked on this for 20-something years. And so we know a lot about the physiological range of the organism. And so in our field measures, we made sure to get enough sites where we could actually be across the breadth of environmental variables. So we weren't truncating any of the response curves, or we weren't only sampling from cold rivers or something. We made sure that they were spread across, and it was very easy for us to do this because we know already where the physiological tolerances for these organisms are. And so for the variables that we chose, we made sure that the populations that we picked were all spread away across the variables. But it's always a problem with these.
Does it scale up? If I wanted to try and test this for a data set sort of on the scale that I have, could I do that?
I don't know. That's my next question. we only have this for this park, but that would be the bigger question. That's what I'm interested in. Is, most of our work is the snail is native to New Zealand, and so we know a lot about New Zealand and the American invasion. But there's also Australia, England, and Asia, and I'd like to be able to build a bigger conclusion across multiple invasion fronts. But I don't know how well these models predict these different responses, and so this was our first test. And so I'd like to be able to go broader scale and make predictions like that.
Okay, if there's no questions, we'll welcome our last speaker of this session, Rodney Mauricio.